Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is our open format portion of the podcast, where we include special topics, interviews, or discussions. My name is Darleen Sandoval, and I'm joined by my podcast co-hosts, Dr. Kirk Haberger and Dr. Kevin Williams. How are you all doing today?
Hey, Dar. Hey, Kevin. Doing good.
Hey, doing pretty good.
Are y'all staying warm?
Trying to. It's been up and down, I guess. Texas, South Texas got a lot of snow, and going over into Florida, I guess I wanted a little bit of it because I ended up going to Chicago, and it was the time that they had negative 25 wind chill.
Oh my
And
God.
I never experienced walking outside and not feeling my ears. Definitely realized you need to get some ear protection and other things when you go into that type of weather. But we escaped it mostly here in Dallas.
Yeah. Kirk, did you all build a snowman?
It was cold. We built
Yeah.
a snowman, and then now it's warm.
Yeah.
Right? We're heading back up towards 70, so instability.
Yeah, it was pretty cold here. We got a bit of snow, but it worked out okay because I got a little bit of acclimatization before I headed to Michigan, northern Michigan, to go cross-country skiing with friends.
Oh,
So
nice.
by the time I got to northern Michigan, it actually felt warm comparatively. So now we're warming up to the 40s, and I'm very excited to run on non-snowed pavement, non-snowed on pavement. All right. Well, Kirk, why don't you tell us what we have in stock for our listeners today in terms of our featured interviews?
Sure. So today we're going to be joined by corresponding author Dr. Francis Lynn from the University of British Columbia for a discussion on his article titled Tracking Insulin and Glucagon-Expressing Cells in Vitro and in Vivo using a Double Reporter Human Embryonic Stem Cell Line. Then we chat with senior author David C. Wraith, Emeritus Professor of Immunology at the Institute of Immunology and Immunotherapy, University of Birmingham, not UAB, and first co-author Michael Price. They'll be talking about their paper, Preclinical Development of a Tolerogenic Peptide from Glutamate Decarboxylase as a Candidate for Antigen-Specific Immunotherapy in Type 1 Diabetes.
We do have a little bit of juggling here. So the paper we're going to discuss by Drs. Rath and Price is actually going to be published in our March issue, and then we'll be interviewing some folks for a paper that's published in the February issue, but they'll come out in the March podcast. So a little bit of juggling in terms of schedule. Kevin, what's happening for our Sweet Talk segment today?
We have Dr. Bret Goodpaster from AdventHealth Translational Research Institute for Metabolism and Diabetes in Orlando, Florida, joining us today. He is here to discuss his 2011 article in Diabetes that is the focus of a Classics in Diabetes article written by Dr. Jeffrey Horowitz in this month's issue. We're going to be discussing the athlete's paradox and the interplay between lipids, exercise, and metabolic health. So stick around to find out more.
Awesome. Let's jump in.
For today's interview, we're going to discuss a featured paper from February's edition. The article is titled Tracking Insulin and Glucagon-Expressing Cells in Vitro and in Vivo using a Double Reporter Human Embryonic Stem Cell Line. We are joined by corresponding author, Dr. Francis Lynn. Dr. Lynn is an associate professor in the Department of Surgery and the School of Biomedical Engineering at the University of British Columbia in Vancouver, Canada. He's also a scientist and head of the diabetes research at the BC Children's Hospital Research Institute. Welcome, Dr. Lynn. Congratulations on your paper, and thanks for being a part of this month's podcast.
Thanks so much for the invitation to come and discuss my work today.
So if you've heard a podcast before, we always like to start with an icebreaker. And so before we talk about your paper, I'd like to know, if you were not in science, is there a career that you think you may have pursued instead?
Well, my other passion growing up was playing the cello. And I started when I was about 6 and continued until well into university. So that was my backup career choice. But I thought science might provide a little bit better income. So that's why I went this route.
Very good.
So, Francis, we like to start by giving our authors the opportunity to highlight any contributing authors or collaborators that were important to this work.
Yeah, so this work was kind of a collaboration and joint effort from a number of people. The work was driven primarily by Samantha Marr, who's a very talented doctoral student in my group. Colleagues from the Canadian Islet Research and Training Network and the UBC Breakthrough T1D Center of Excellence were also instrumental in supporting this work. And then, I'd like to also thank the reviewers and editor for at Diabetes for their excellent suggestions, which really helped us get this manuscript over the finish line.
Yeah, we always talk about how science is such a team sport, right?
Mm-hmm.
All right, so Dr. Lynn, maybe you could start with an overview of your research and how this project kind of grew out of that overall focus.
Yeah, so my research program focuses on how islet cells form during embryogenesis, how they mature and become functional after birth, and then how they kind of function in the adult. And sort of the overall translational goal of the work is to bring a functional cell-based therapy to the clinic for those living with diabetes. So when I started my lab, we mostly used mouse models to do this type of work. But over the last 15 years or so, with the advances in our abilities to differentiate stem cells into islet cells, I think we now have a pretty powerful tool to study human islet development and function in the lab. So this particular project, over the last 10 to 15 years, it's becoming kind of increasingly clear that alpha cell dysfunction also contributes to the etiology of both forms of diabetes, or both of the major forms of diabetes. And so Samantha's thesis focuses on a transcriptional co-regulator in alpha cells. And her work suggests that this protein plays a key role in the maturation process in alpha cells. And that process is relatively unexplored. And so I guess the main impetus for this work was it's really difficult to get enough alpha cells out of a mouse to do any kind of molecular studies. So Sam thought, why don't we make an alpha cell reporter that would allow us to purify large numbers of non-tumor derived human alpha cells so that we could do these molecular studies. Along the way in developing this cell line, we realized that we didn't really have a good grasp of kind of when the alpha cells were specified or differentiated during our protocols, and therefore what the best time in that differentiation process would be to take those alpha cells for our work. The study really allowed us to, develop that cell line so we could use those alpha cells for something else, and then understand the differentiation of alpha cells along the way. So that's kind of how we got to making this cell line.
So let's talk a little bit about that, about how you established this cell line, and importantly, some of the validation work that you did. So I guess I should back up and specify that these two cell lines are basically distinct cell lines that have, so basically an alpha and a beta cell line that have specific fluorescent reporters to each cell line, correct?
Yes, it's similar to that. So this is one cell line that has two fluorescent reporters that tells us when insulin gene expression is active and when glucagon gene expression is active. How we kind of got to that cell line is, I guess, for the last 12, 13 years, we've been doing CRISPR editing in pluripotent stem cells. And the approach we used is we first put a fluorescent reporter downstream of insulin in a pretty standard embryonic stem cell line. And then once that was done, we took that reporter cell line and we put a different fluorescent reporter downstream of glucagon. And then we, did the studies that were in this paper. And so basically, once we have that cell line, and I guess taking a step back, something that was kind of unique about this study is, compared to past studies, was that the modifications we made didn't disrupt the hormone expression in either of these loci. That means that we should have, normal hormone expression and secretion. And so once we had the cell lines generated, we did the standard tests we do. So we see if the cells can differentiate into the 3 different germ layers. That kind of gives us some confidence that they're what's called pluripotent. That we looked for karyotypic abnormalities that might impact differentiation or function. And then we checked to make sure they could, in general, differentiate to endocrine cells as efficiently as the cell line we started with. And all of those things seemed to be fine. And then we were kind of ready to go and assess how well those fluorescent reporters reported on insulin and glucagon expression. In order to do this, we first did some immunostaining to look at the overlap between our fluorescent reporters and the insulin and glucagon genes. And then we used flow cytometry to quantify that overlap. And then we used fluorescent FACS, or a sorting protocol, to separate those populations and look at gene expression within them, and found that those fluorescent reporters faithfully kind of reported an alpha and a beta cell phenotype. And finally, we kind of took those cells and showed that they could secrete insulin and glucagon both in vitro and in vivo. And then we used some intravital imaging on cells that have been transplanted into the interior chamber of the eye of mice to look at whether we could image those cells in vivo. Kind of in sum, we did a lot to characterize the cell line, kind of with the hope that others would be confident that it would be of use to them. And since we published this work, we've had a number of requests for this line, suggesting that we were somewhat
Convincing.
Suggesting
Absolutely.
that what we presented was somewhat convincing to others. So we're pretty happy with the response so far.
Excellent.
Francis, you kind of alluded to this in your introduction there, but the overall goal is to take stem cells, derive them into islets, and use them therapeutically for people who are insulin insufficient, right? Tell us about the current obstacles that prevent us from doing that already, and then how you think your work here may bring us potentially closer to that goal.
I think the potential for a stem cell-based therapy for type 1 diabetes, and then maybe all forms of diabetes, has really come a long way over the last 10 years. And maybe the listeners will know, but there's quite a large number of pretty promising clinical trials currently underway. Despite that promise, I think the field still wrestles with a couple pretty big obstacles. And the first is preventing the immunorejection of that transplanted tissue. So similar to what we have to do for any transplantation procedure. And the second is improving the transplanted islet cell kind of resilience and then function. So my research program has interests in kind of both of these areas, but I think probably the bigger focus currently is improving the function and resilience of the stem cell-derived islets. So on the resilience side of things, your listeners may know that transplanted islets face a huge number of stresses. So immediately following transplantation, there's hypoxia. Beta cells need a lot of oxygen, so that's stressful. There's ER stress, so the need to produce hormones is stressful for the islet cells. And then there's inflammatory stresses from both the innate and adaptive immune systems. And there's also a whole bunch of other stresses that the cells face. So we're kind of interested in developing approaches to mitigate those stresses and make the cells more resilient. And then on the function side, which is maybe more where this manuscript fits in, the cells that most labs produce now from stem cells do not function equivalently to human islets. And the result of that is a dampened secretory response. So they don't respond to glucose in the same way that a human islet cell would. And so that's a large focus of our current work, is trying to understand the genetic and physiological mechanisms that help those embryonic or neonatal islets that we think we derive from the stem cells become functional. And that process is called maturation. So this published work kind of helps us towards that goal, because now we can separate stem cell-derived beta cells, stem cell-derived alpha cells, and study those cells and understand the pathways that are important for that maturation and function. And so I should probably just note that cell therapies aren't the only kind of potential use for these types of cells now. Because we're getting so good at making the cells, we can now think about using stem cell-derived cells for disease modeling, for instance, trying to understand how genes that might predispose to diabetes impact beta cells, and also probably just developing better understanding of islet biology in general. So I think it's a really exciting time to be an islet biologist with all these new tools and genome editing and the like.
Yeah. So your study also uncovered an interesting propensity of the bihormonal cells to drift towards an alpha cell phenotype. Can you tell us a little bit about those findings and what you think this means moving forward?
Yeah, so we kind of think this is a pretty interesting finding reported by our work, and maybe not totally novel, but others have kind of made the observation in the past that these polyhormonal cells likely become alpha cells as differentiation proceeds. However, to the best of our knowledge, nobody has demonstrated that these cells directly, in kind of a cell intrinsic or cell autonomous manner, become alpha cells by sorting them away from the rest of the cells and differentiating them in isolation. And so that's what we showed, that these cells transition from this polyhormonal fate to a monohormonal fate directly over time without any input from any other cell types in the culture, or without any, we think, input from whatever we're adding to the culture. And so we think this was an important observation to make because it kind of gives us a good understanding of when during differentiation insulin expression becomes a true marker for beta cells, and also when we can expect insulin to be lost from alpha cells. And so just in us thinking about, when we're going to take these stem cell derived alpha cells for future study, this gives us a good understanding now of when we should do those types of experiments. So yeah, that's kind of the observation that we made about the polyhormonal cells. So I guess going forward related to this observation, we might want to determine whether we can speed up the transition from a polyhormonal cell to a more monohormonal alpha cell and how that process works. So we could use some high content screening approaches for that. We also might want to understand whether we can force those polyhormonal cells that normally become alpha cells to become beta cells, because then we could get a more uniform mixture of beta cells in our differentiations. So that's kind of some ideas about where we think that observation could go next.
Yeah, very, very cool. Francis, are there any other key points or thoughts that you want to leave us with as we close the interview?
I think it's just a really exciting time to be doing islet biology and stem cell research, as I said, and I look forward to seeing more and more studies like this published in diabetes. Thanks again for your interest in our research and for highlighting us in diabetes bio.
Absolutely. We want to thank you for joining us today and for sharing your expertise and discussing your recent work on the human embryonic stem cell derived pancreatic alpha and beta cell line. We think that it's an exciting time as well, and beautiful, beautiful images, by the way. Those I'm so jealous of you islet biologists and the IFs that you put out there. So I encourage the listeners to read the full article in the February issue of Diabetes. And thanks again for joining us.
Thanks so much.
The next feature article we will discuss today is titled Preclinical Development of a Tolerogenic Peptide from Glutamate Decarboxylase as a Candidate for Antigen-Specific Immunotherapy in Type 1 Diabetes. We are joined by co-lead author Michael Price, who is a research fellow in Dr. Wraith's group, and Dr. David Wraith, an Emeritus Professor of Immunology. Both are at the Institute of Immunology and Immunotherapy at the University of Birmingham in the UK. Welcome, Drs. Price and Wraith. Congratulations on your paper, and thanks for being a part of this month's podcast.
It's great to be here. Thanks for having us.
Great pleasure.
So we like to start our conversation with a little bit of an icebreaker. So, and I know, Dr. Price, you're still early in your career, but if you weren't in science, what career do you think you would have pursued?
I think I would probably be a chef. I do love food and I
Nice.
love cooking. So it was, I've always been told by my parents that, and my fiance, that I should have been a chef, wasn't a
Nice.
scientist.
I feel like, with molecular biology, you're following recipes anyway, so very similar.
That's right. What's the difference between a protocol and a recipe?
That's right.
I've been told.
Dr. Wraith, what about you?
Probably a farmer, country boy.
Nice.
Yeah.
So, Dr. Wraith, we like to give our authors an opportunity to highlight any other contributing authors or collaborators that really helped with this work. Is there anyone in mind that you'd like to start with?
Yeah, I mean, the lead co-authors on the paper were Mike and Sky, and Sky and Mike have really driven this project together. Some of the work on the really important work on the assay that we developed through this was done by Naomi Richardson, who was a student in the lab as well. So those are the other two I'd like to highlight.
Wonderful. So I guess moving on, maybe start by giving us an overview of your research program and then how this specific project grew out of that focus.
Okay, so our work focuses on antigen-specific immunotherapy. And this work dates back to really to the 1980s when I was working over in California at Stanford University. And we found working there on models of multiple sclerosis that synthetic peptides could be used to selectively switch off the CD4 T cells that you all know are the central cells orchestrating the immune response. So we've really been working on this ever since then. And over the years, we've found ways of delivering synthetic peptides that's both safe and effective. And we've defined the rules on how to design effective tolerance-inducing peptides. And our real aim is to reinstate self-tolerance in different autoimmune diseases. And I guess over the last 10 years, our work has really defined transcriptional and the epigenetic changes that govern antigen-specific induction of tolerance and generation of regulatory T cells. And over the years, we've tried to apply this into the clinic and have developed therapies for Graves' disease and multiple sclerosis. And these have both been through phase 1 and phase 2 clinical trials with good evidence of safety and preliminary, of course, evidence of efficacy. And it was really through association with one of the endocrinologists working with us that, we decided to apply all this to type 1 diabetes. So our real aim is to design a set of peptides that could be used, if you like, to prevent development of type 1 diabetes. So, Mike has really been involved with this type 1 diabetes program. So I think it's only fair for him to describe it to you.
Well, that's great. So, Dr. Price, why don't you tell us a little bit more about the rationale for this specific project and maybe explain, especially to our non-immunotherapy folks, the significance of developing an antigen-specific immunotherapy for type 1 diabetes as opposed to non-antigen specific approaches?
Sure. So, as I'm sure all of your listenership are aware, the anti-CD3 therapy to plutamab had recently been approved, and that's the first sort of real therapy for type 1 diabetes since insulin, but it's still non-antigen specific. It's not only targeting those T cells that are attacking the beta islet cells and the antigens that are being exposed there. So the real driver for antigen-specific immunotherapy is that we want to restore tolerance to these antigens in a targeted way whilst leaving all other T cell responses unaffected, which these non-specific immune modulating therapies can do. So following on from the rationale behind David's work and why we chose the work on the type 1 diabetes is that there are a number of well-characterized antigens in type 1 diabetes that the T-cell responses are generated towards. So one being glutamate decarboxylase, or GAD65. So from there, the rationale was that we used predictive algorithms to design candidate peptides that were predicted to bind to multiple HLA alleles that are associated with type 1 diabetes. So these are highly associated with patients living with type 1 diabetes. For example, the HLA DR04 allele or the DR03 allele. And from there, these predictive algorithms gave us a list of candidate peptides. And according to David's work, previous work, these were extended out to larger peptide molecules to encourage antigen processing and so that we can identify natively processed epitopes. So from there, really the start of the work was to figure out which of these peptides actually can induce responses in the T cells from patients in a selective manner over non-diabetic controls.
Thanks for that introduction and maybe digging in a little deeper. Michael, can you tell us how you applied the principles of processing independent T cell epitopes to type 1 diabetes, and maybe also why glutamate decarboxylase 65 was the target you chose?
Yes, great question. So we also, in our lab, we have been working on targets to other type 1 diabetes proteins, so proinsulin and IA2, but that work isn't as far ahead as along the line as the work on the GAD peptides. So at this point, we've obviously got to this stage with the GAD-P10 peptide. And so addressing the first point, so applying the principles of tolerance-inducing peptide design to the P10 candidate that we'd identified, from the stage that we'd had shown that the P10 peptide selectively induced stronger responses in to PBMCs from patients with type 1 diabetes. We then took a HLA transgenic mouse model, which expresses the HLA DR401 allele. And from there, we immunized that transgenic mouse with the P10 peptide to generate antigen-specific T-cell hybridomas. With these, we can elucidate the minimal core epitope that is within that peptide 10 or the P10 30mer. And we can show by responses of the hybridoma to whole GAD antigen that this epitope is natively processed, which is a key property of tolerance-inducing peptides. So from there, we identified this minimal epitope, which we called 10.5, and then we applied the remainder of the key principles of designing these tolerance-inducing peptides. So the first is that you need the peptide to be soluble. So by that, we added different numbers of lysines or different amino acids to increase the solubility of the peptide. We tried out these iterations around the peptide and found that the soluble derivatives could still generate responses in the hybridomas. The next principle to address is that the candidate tolerogenic peptide needs to be able to, it can induce a response independent of antigen processing. So for this, we used antigen-presenting cells that are still live but have been fixed a formaldehyde treatment, so that the peptides are still able to bind into the HLA class 2 on the surface, if they're able to. And we found that the soluble P10 derivatives could do so. And then the last principle is that the soluble P10 derivative is able to bind to steady state dendritic cells in vivo. So for this, we re-immunized the HLA DR4 transgenic mice with the soluble peptide, and very quickly after immunization, we took the splenocytes and used those as antigen-presenting cells, either by themselves. So the only way that they could have seen antigen is if it would bound to the empty class 2 on the surface, or you gave back exogenous antigen. And from that set of experiments, it clearly showed that the P10-sol derivative could bind the steady state dendritic cells and thus met the last criterion of being a tolerance-inducing peptide. And from there, we took it forward to further tests where we test its tolerance-inducing capability via this dose escalation protocol.
That was a really nice explanation, or sort of step by step, of the results that you have found in this paper. And one of the things that I wanted to talk about that I thought, I think whenever a paper can do this, it's really cool. And that is using the combination of preclinical and clinically targeted methods. So, what are the challenges and limitations with, in your work particular, of this combination of preclinical and taking the preclinical research to clinical therapy? And what are the steps to overcome some of these obstacles?
So in this work, we identify the minimal epitope P10 from P10S, or P10-sol, sorry, using this approach of preclinical HLA transgenic mice, which there are some caveats of using, but importantly, using the dose escalation tolerance-inducing protocol, we show that the soluble P10 epitope does induce tolerance to the whole 30mer. And after that, we retest this P10 soluble epitope in a further cohort of people with type 1 diabetes. So we take PBMCs from them, and that we show that despite this epitope being characterized using a preclinical murines mouse system, it's actually a patient-relevant epitope with PBMCs from people with type 1 diabetes can respond to. So really emphasizing that it is patient-relevant. And the sort of a limitation that we've tried to overcome towards this is by developing this new fluorescent dye dilution and activation induced marker assay during the course of the work, is that we want to move towards a human-centric workflow where we discover the epitopes from the autoantigens entirely using human PBM or patient PBMCs, and then doing all of the minimal epitope and the tolerance-inducing peptide criterion meeting in a fully human workflow. And that is it's not to say that this epitope that we've report here doesn't meet those criterion, because we showed that it has and that it is responsive in patients, but moving forward, that's what we are going to start applying in our novel workflows.
Excellent.
David, I wanted to return to you and just get your critical thoughts on the overall implication of this work, and maybe summarize its importance in the field.
Thanks for that, Kirk. I mean, I think, any identification of potentially tolerogenic peptides is incredibly important in any autoimmune disease. But the implications of this work is that, given that we were able to show that this designed peptide would induce tolerance, at least in the HLA-DR4 transgenic mice, based on our previous work in MS and Graves' disease, we would fully expect this peptide to induce tolerance in humans and in patients with diabetes. So, we are actually in the process currently of testing this peptide in, if you like, phase 0 clinical trials with collaborators in the UK. So I think, if we are able to demonstrate this, we aim in the future to identify more epitopes, as Mike said, from proinsulin, from IA2C, and we've identified other regions of GAD, which are extremely potent stimulators of responses in patients. So we want to design additional epitopes and then produce really targeted precision medicine treatments for patients. Ultimately, of course, our dream is to be able to identify even children when they develop those one or two anti-islet antigen antibodies and start treating them with a peptide formulation that is completely safe, really to stop the disease in its tracks and stop it before it really develops. That's the ultimate aim.
Yeah, that would be amazing. Doctors Price and Wraith, we want to thank you both for joining us today to share your expertise and for discussing your recent work focused on the antigen-specific immunotherapy in type 1 diabetes. I encourage listeners to read the full article in the March issue of Diabetes. Thank you.
Thanks, guys.
Thank you both. Thank you all.
Thank you all.
Welcome to another edition of Sweet Talk. Today we're excited to welcome Dr. Bret Goodpaster, a leading figure in exercise physiology and metabolic research. Dr. Goodpaster is the scientific director and senior investigator at the AdventHealth Translational Research Institute for Metabolism and Diabetes in Orlando, Florida, and also holds a joint appointment at Johns Hopkins University as professor of medicine. He has made seminal contributions to understanding the interplay between skeletal muscle metabolism, exercise, and insulin sensitivity. His work on the athlete's paradox, published in Diabetes in 2011, challenged long-held assumptions and opened new avenues for research. Dr. Goodpaster has also been a driving force behind numerous studies examining how diet and physical activity interventions can improve cardiometabolic risk factors, as well as metabolic health in individuals at risk for type 2 diabetes. Thank you for joining us, Dr. Goodpaster.
Hey, Kevin, good to be with you.
Before we dive into the discussion, we always like to start off with a little bit of an icebreaker. If you could solve one answered question in diabetes or obesity research today, what do you think that would be?
Yeah, it's a great question. I would say, virtually all of my research since graduate school has been focused on why exercise is beneficial and how it works from a mechanistic viewpoint. But I also recognize that it's really important to understand, what motivates people to exercise, because I think we would all agree that if we could get more people to exercise, we would solve a lot of our problems around diabetes and cardiovascular disease risk. So I would really like to understand better what's the brain health connection to what motivates people to exercise, what makes some people feel good when they exercise and some people don't feel good after they exercise. And I think if we could unlock some of those mysteries around exercise and mood and motivation, I think we'd solve a lot of these problems about diabetes.
Yeah, it's really fascinating, the motivational aspect and that reward or wanting to come back and exercise again.
Exactly.
So yeah, it's really interesting.
And from a broader perspective, let's talk about the field. So in 2011, there was a lot of debate about the relationship between intracellular lipids and insulin resistance. Could you talk a little bit about the sense of the state of the field at that time and the debate about exercise influenced it? And let's even get into a little bit of the athlete's paradox.
Yeah, Darlene, I would even go back before that. So when I was a early stage, faculty at the University of Pittsburgh, we were interested in, metabolic flexibility and the effects of exercise on the ability to utilize fatty acids for energy and storage of fatty acids. It was known at that time that athletes stored muscle triglyceride as a fuel source. And we were doing work with, David Kelly and insulin resistance and diabetes risk. And we did a weight loss study and showed that intracellular lipid content decreased with weight loss. And that was seen in conjunction with their improved insulin sensitivity. And that was really in line with what the field was, thinking at the time. It was almost becoming dogmatic that, muscle triglycerides were a cause of insulin resistance. And our weight loss study supported that. So we were also interested in the effects of exercise. So before we did an exercise training study, which obviously takes a lot of time and effort and money to do these types of intervention studies, we brought in a group of highly trained endurance athletes in the lab, and we did glucose clamps to measure their insulin sensitivity, and we did muscle biopsies, measured the intramuscular lipids. And our hypothesis, and it was borne out in this study, was that, and this was based on work that Hans Hoppler did in Switzerland in the, I think it was in the 80s, showing that highly trained athletes had more triglyceride in the muscle. So this was the athlete's paradox, that they had more, our athletes had more muscle triglycerides, but they were very insulin sensitive. And I think this really helped the field sort of change the mindset to start really looking at other muscle lipids and muscle lipid metabolites, like diacylglycerol and ceramide. And I think that was a paper that I feel that was important, because while it didn't necessarily infer cause and effect, it certainly told us what likely was not causing insulin resistance. And I think that was really important.
Yeah, building on that, I think, you alluded to it a little bit, that an important part of the study was not just the total amount of lipids, but their specific molecular species and subcellular localization. Could you elaborate a little bit more on why that distinction matters, how it framed or reframed the conversation around lipotoxicity and insulin resistance?
Yeah, and I think that was another, part of the evolution of this whole story. Like Darleen said, in 2011, we published, kind of revisiting the athlete's paradox with my PhD student Francesca Amati, and we studied older athletes compared to older, age-matched sedentary people. And this time we not only measured muscle triglycerides, but also muscle ceramides and muscle diacylglycerol, because again, in 2011, it was thought that, okay, muscle triglycerides aren't causing insulin resistance, so it must be these other lipids. There were lots of animal studies at the time that supported a role for either diacylglycerol or ceramides in insulin resistance. And that was really sort of two divided camps. So we thought, well, let's, kind of take an unbiased approach in a human study, to see what might be related to insulin resistance. And we saw that ceramides were related to insulin resistance in that study. Diacylglycerol, it became a little bit more complicated. Some diacylglycerol moieties were associated with insulin resistance, and some were actually associated with better insulin sensitivity. So I think, this just added another layer of complexity to the story. Which type of lipid was it? Where, did it matter where it was located in the cell? In that study, we didn't do that, but other studies subsequently have started to look at, sort of subcellular localization of these lipids. So it became really complicated.
So, can you talk a little bit about the findings along the way that really surprised you? And you mentioned some of these in your actual classics article. So, maybe highlight some of those particular findings.
Yeah, I think the surprising findings were really around the diacylglycerol. We actually showed that, and this was contrary to our hypothesis, that some of the most prevalent diacylglycerol species in skeletal muscle were actually 2 to 3 fold higher in these endurance trained athletes, not lower. Yeah, it was like the athlete's paradox, except with diacylglycerol. Diacylglycerols were higher in endurance trained athletes. But then when you dive deeper into these different molecular species, some of the species were actually, were higher in insulin resistance. So again, this was the surprising finding to us, was that, not all these diacylglycerols were the same. And I think others like Brian Bergman and Jerry Schulman started to really dive deeper into these diacylglycerols and ceramides, and where they were in the cell, and how they were implicated in insulin signaling. And I think there was a lot of great work done by them in that space. We just sort of made some observations that, in a human study, associations, but nonetheless, I think were important to show perhaps what wasn't necessarily cause and effect.
So, to layer a little more complication onto that conversation, how do you think diet interacts with those findings? Or do you know?
Yeah, I mean, diet is a complicated thing, right? It's not only total energy balance, calories in, but also the composition of the diet, low fat, low carb, the type of fat you're eating, etc. We haven't done a lot of those studies, but I do think it's really, really important. I think diet composition probably matters with respect to, what type of lipids accumulate and which ones don't. And I don't know if there's that much great data on diet composition and these muscle lipids, to be honest.
I'm curious too, you started off early on in your career, you mentioned trying to understand metabolic flexibility and the role of these IMCLs in obesity versus the exercise endurance. I was curious, is there anything known about the flexibility of the transition between these different states? How long of an exercise does it take? How much endurance, how much training would it take in order for the IMCLs to potentially be beneficial versus the negative effects that take place in an obese state? Is there anything that's known about facilitating that shift in how that takes place?
Yeah, that's a great question. The effects of one bout of exercise are very powerful in terms of, stimulating fat oxidation. Most people don't really appreciate that, just during moderate exercise, you're oxidizing during just moderate intensity exercise, sometimes a half a gram per minute of fat. I mean, that's, over 60 minutes, that's a lot of fat that you're burning. And I think training the muscles to oxidize fat better is certainly going to promote, again, greater fat utilization from plasma sources, adipose tissue, lipolysis, but also those stored muscle triglycerides. So I think the big question moving forward is, like you point out, Kevin, the dynamics of exercise and exercise training and the fat, the intramyocellular lipid turnover in the muscle. If the muscles aren't being oxidized very much, and if they just sit there, do they turn rancid or something? Do they turn bad? Do they change their composition? I don't think anybody really understands that very well.
So fast forward 10 years, what problems would you like to be solved in terms of these specific questions?
Well, it's interesting because, a lot of us are still interested in this so-called target of skeletal muscle insulin resistance, right? And currently we don't have any drugs that treat muscle insulin resistance. And not only related to diabetes, but I think related to pre-diabetes and diabetes risk, cardiovascular disease risk, independent of diabetes. I think insulin resistance is likely an important target. So, the big question is, can we identify targets in these lipid storage or lipid oxidation pathways in muscle that directly relate to improved insulin sensitivity? And will that, mitigate or mediate diabetes risk and risk for, cardiovascular disease risk, metabolic syndrome, et cetera? I think that's what I would like to see, happen out of all this, because otherwise it's interesting, but if you don't really have any targets for intervention, you could argue that it's not as important.
Perfect. Thank you, Dr. Goodpaster, for joining us today and sharing your research and insights into exercise, lipid metabolism, and metabolic health. And thank you to our listeners for tuning in to this episode of Sweet Talk. Before we go, Dr. Goodpaster, could you let the listeners know where they might be able to learn more about your work? Do you have a website? Are you on the socials?
Yeah, I have a LinkedIn profile. You can search under my name. It's a fairly unique name, not that difficult to find. And as you pointed out, I'm at the AdventHealth Translational Research Institute in Orlando, Florida. Drop me a line. I'd be happy to have a chat with you.
We also want to remind our listeners to check out the Classics in Diabetes article in Dr. Goodpaster's 2011 study, which was titled Mechanistic Insights into the Exercise-Induced Changes in Muscle Lipids and Insulin Sensitivity, Expanding on the Athlete's Paradox, which is featured in this month's issue of Diabetes. As always, don't forget to like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes. And are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.